Hello and welcome to Lesson 6 of Unit 1 of the Higher Human Biology course. This lesson is going to be focusing on Topic 6, which is Metabolic Pathways. So this is a tricky topic. It's one that students often find is one of the most difficult when it comes to the exam. It's also quite heavily examined, so you tend to find a question on it every year in the exam. And it's usually an applied context type question where they try and apply it to a new or unfamiliar context for you. So these can be quite tricky ones. So we'll need to make sure that we go over things thoroughly on this topic and that you get a good understanding of it by the time that we've finished and you've gone over and did a bit more of your own studying as well. So, what are metabolic pathways? Well, metabolic pathways are a series of reactions which happen within cells within the body, and those reactions can be involved in many of the major processes that our cells undertake, such as aerobic respiration and protein synthesis that we have learned about so far. Metabolic pathways they're integrated and controlled pathways of enzyme catalyzed reactions within the cell. So they're integrated. That means that all the steps happen within the cell. They're controlled by enzymes. Okay, they are enzyme catalyzed reactions. Now, when we were in National 5 biology, we learned about the likes of respiration and photosynthesis as enzyme controlled reactions, and these are a good example of some metabolic pathways that we already know about. Okay, so they're controlled by enzymes, which means that the concentration of enzyme will have a big impact on how fast these reactions go because, as we know, enzymes do speed up these reactions. Now, metabolic pathways can have reversible steps, irreversible steps, and alternative routes. And if we have a wee look at our diagram here, we can see some examples of those. So this here, the substrate has been broken down into a molecule. We'll just call it intermediate 1 for the sake of this. Now, that is an example of an irreversible reaction because the arrow is only going in one direction. The breakdown of intermediate 1 to intermediate 2, it does go this way, but it also goes back the other way as well. So we call this an reversible reaction. Okay, it's a reversible reaction. So it can go both ways. If we have a high concentration of intermediate 2, for example, we can convert some of that back to intermediate 1. Okay, so that's reversible. And we also have this alternative route as well. Now, that will probably only come into play really if, for example, let's say this enzyme here, for whatever reason, stops working. Okay, and that will cut off this part of the pathway. The substrate, in order to get to the product, will have to go via an alternative route. Okay, and there's usually another intermediate product here that the substrate will break down into, and then that intermediate product will break down into the end product. Okay, and we see that in the likes of aerobic respiration, amongst other things. Now, reactions within the cell can either be anabolic or catabolic. Now, at National 5, we learned about synthesis and we learned about degradation. Now, anabolic and catabolic reactions are very similar to our synthesis and degradation reactions we already know about. And we can see just at the bottom of the screen here, we've got our anabolic and our catabolic reactions here. And we can see that the anabolic reaction here is building up from larger molecules, two larger molecules from smaller molecules. Okay, so we get our small molecules here. We are putting in energy. This requires energy. And we're getting our larger molecules here, okay, from small to large requiring energy. Over this side here, we've got the catabolic reactions taking place. That involves the breakdown of our large molecules into smaller molecules, and this reaction releases energy. Okay, we can see that there, large molecule all chained together. It is broken down, it releases energy, and we get all these small molecules as products at the end. Okay, so you need to remember which of these reactions is which. If you think about anabolic a bit like the synthesis reactions we learned about at Nat 5, and catabolic like the degradation reactions that we learned at National 5 as well. Okay, so hopefully all of that makes sense. Metabolic pathways controlled by enzymes. We have reversible, irreversible, and alternative routes, and our reactions can either be anabolic or catabolic, and we need to know the difference between those two for our exam. Okay, So, metabolic pathways are controlled by the presence or absence of particular enzymes and the regulation of the rate of reaction of key enzymes. So, what does that mean? Well, if the enzyme is present, the reaction and the pathway are therefore going to proceed and they're going to go faster. If the enzyme is absent, then obviously that pathway will come to a halt. And if we want to, for example, slow down a reaction. So people often talk about our metabolism. Now, our metabolism is controlled by the sum of all of these reactions that happen in our cell. So the more of these reactions that are happening, the greater our metabolism or the higher our metabolism. The less of these reactions that are taking place, the lower our metabolism. Okay. And that is all controlled by these enzymes. If they're in higher quantities, our metabolism is going to be greater. If they're in lower quantities, our metabolism is going to be lower. Okay. So how do enzymes actually carry out their reactions? So a quick recap on NAT5. Now we learned at NAT5 that on the enzyme we have an area called the active site, and that active site binds to a specific substrate. Okay, and that's really as far as we went at National 5. The shape of that active site was complementary to our specific substrate, wasn't it? Now, in an ideal world, that would be perfect and it would be tie that in a neat little bow, and that's how we'd understand it, but it's not quite how it works. Now, when the substrate does enter the active site, the substrate doesn't have quite the same shape, okay? The shape is just slightly different, okay? It's very similar, but it's just slightly different. Now, what needs to happen then is for the active site actually needs to change shape, okay? The active site changes shape, okay? And the reason it does this is to better fit the substrate after the substrate binds. And this process is called induced fit. Okay, and we can see it in the animation over here on the right. The substrate enters the active site, but when it initially enters, the active site isn't quite snug on that particular substrate, and therefore it has to change shape to better fit the substrate. Now, induced fit is good for reactions because it lowers the activation energy. That's the amount of energy that is required for the reaction to get started. Okay, and obviously, with a lower activation energy, that's better for cells because they can start the reaction sooner and by using up less energy. And that's going to be good in terms of speeding up our metabolic pathways. Okay, so induced fit lowers that activation energy required for the reaction as the substrate is more closely bound to the active site. Now, the substrate molecule has a high affinity for our active site. That means it has a high level of attractiveness for the active site. It wants to bind to it. Okay, so two molecules with a high affinity want to bind with each other. All right. Now, once the reaction has taken place and we have our products, those products leave the active site because they have a low affinity for it. Okay, products have a low affinity for the active site, which is why they are then able to leave. That's why when the substrate goes in, that molecule can eventually leave because when the reaction's taking place, the products have a lower affinity. Okay, so affinity, that's the attractiveness between two molecules, and the substrate has a high affinity for the active site. The product has a low affinity for the active site. Quick shout out, as always, to the Amoeba Sisters. They've got a nice little video that shows nice animations of how enzymes function and how induced fit functions. So if you want to see that, you can go and subscribe to their channel and watch the video. Okay, so what other factors can affect the rate of enzyme reaction? How fast that enzyme goes, the enzyme activity, to use the term that we used in National 5. Well, they can be affected by a few things. The first thing we're going to have a look at is something called the substrate concentration. Okay. And simply put, that is the concentration of the substrate molecule that binds to the active site. If we have lots of that substrate, we're going to have more enzymes binding to the substrate and therefore more enzyme activity. Okay, the rate of the reaction will increase. And we can see that in our little graph here. We have the enzyme activity increasing here, and the graph continues to go up as the substrate concentration also increases. However, we reach a point round about here on the graph, okay, where the enzyme activity starts to level off. Okay, now at this point, continuing to add substrate actually makes no difference to the reaction rate. And the reason for that is because all of the active sites that are on our enzyme are occupied by substrate. Okay, so it's like 20 students walking into a classroom and there are 20 seats. Okay, now initially pupils will be able to find a seat, and those seats will fill up and fill up and fill up, and we'll have more and more students seated. But then if we continue to just keep adding students to the class, once those 20 seats are occupied, it doesn't matter if we keep adding students to the class, they won't be able to have a seat and they won't be able to sit down for the lesson. Okay, and it's a bit like that in terms of enzymes and substrate. We can keep adding substrate all day long, but because all the active sites are occupied, it will make no difference to enzyme activity. Now, some metabolic reactions are reversible. We spoke about this earlier on, okay, that word reversible, which means that the product can be reverted back to the substrate. Okay, we can reverse that reaction. And the presence of a substrate or the removal of a product can drive a sequence of reactions in a particular direction. So, for example, in something like aerobic respiration, the presence of glucose will drive aerobic respiration. Okay, it will drive that series of reactions that occur to produce our ATP at the end. And similarly, if you remove a product, that may well stop that metabolic pathway, and we'll have an accumulation or a build-up of a particular intermediate substance in the middle of the pathway, unless it's reversible, of course, in which case it can go back to the initial substrate. Now, inhibition is the last part of this topic, and there are three main types of inhibition you need to know about for your exam, the first of which is competitive inhibition. Now, competitive inhibitors are inhibitors which bind at the active site, okay, and by doing so, prevent the substrate from binding. So if we have a look here at a normal reaction, we've got the enzyme here, we've got the substrate here, and the substrate's able to enter the active site, and we get an enzyme substrate complex. Okay, all good. But when this red inhibitor here is present, there is now a 50-50 chance that that inhibitor will reach the active site before the substrate does. In which case, if it does, it will block the active site and prevent the substrate from binding, which means there's going to be no reaction on that enzyme. The way that we overcome this, the way that we get around this competitive inhibition, the way that we reverse it, is by increasing the substrate concentration. And if we look at our diagram to understand that, it's, for example, we keep the inhibitor just at 1 there, but we make the substrate 4, okay? So we've increased the substrate concentration. That now means that instead of being a 50-50 chance that the substrate will bind, there's now an 80% chance that the substrate will bind first, okay? The inhibitor's got a 1 in 5 or a 20% chance of binding, but the substrate has got an 80% chance. So therefore, if we keep adding substrate, we're going to eventually overcome the inhibitor, okay? Because we're lowering and lowering the inhibitor's chance of getting there before the substrate. So the reaction rate, the enzyme activity will continue to increase. The second type of inhibitor we need to know about are non-competitive inhibitors. Now, those ones are slightly different because they bind away from the active site, not on the active site. But when they do that, when they bind away from the active site, as we can see in the diagram here, the non-competitive inhibitor changes the shape of the active site, so it's a kind of square shape here initially, and it changes it to this sort of V shape. And what that means is the substrate is not directly competing with the inhibitor, but because of the inhibitor's action, the substrate can no longer bind to the active site because the shape has been changed, and therefore the substrate can't bind to it anymore. Okay, so a non-competitive inhibitor binds away from the active site and it changes the shape of the active site, which prevents the substrate from binding. Now, a non-competitive inhibitor cannot be reversed by increasing substrate concentration. And we can see from our graph here, the non-competitive inhibitor, no matter how much substrate we add to it, the non-competitive inhibitor does not increase enzyme activity there, okay? The graph just stays level, so it remains low throughout, okay? And we can compare that to the competitive inhibitor, which eventually, with added substrate, will overcome inhibition, and compare that to the initial no inhibitor graph, where we see the enzyme activity increasing until all the active sites are occupied there. And lastly, feedback inhibition. Now, feedback inhibition is a little bit different. Feedback inhibition is to do with the end product. Now, if we go back to the pathway that we had earlier on, this metabolic pathway here, we can see a substrate being broken down to an intermediate, another intermediate, and then the end product. And there are 3 enzymes that help us to catalyze those reactions. So feedback inhibition is when this end product, okay, that's this thing here, the end product, in the metabolic pathway reaches what we call a critical concentration, okay? essentially at the point where we don't need to make it anymore, because it starts becoming a waste of the cell's resources and energy to keep making it because the cell doesn't require it. So when it reaches that critical concentration, the end product itself will then move back and it will find an earlier enzyme, okay, in this case, enzyme 1, and it will inhibit that enzyme, okay, so it inhibits that earlier enzyme. And what that means is that it will block that pathway, okay, so that the substrate can no longer be broken down, and therefore the reaction and the metabolic pathway will not continue, okay. It prevents further synthesis of the end product, okay. This will not be produced because none of the pathway will proceed, and the substrate is no longer able to be broken down into the intermediate because this enzyme here, the first enzyme, is being inhibited by the end product. Okay, so we call that feedback inhibition because the end product is going backwards in the pathway and inhibiting that first enzyme. Okay, we're going to stop the lesson part there and we're going to go and have a look at the past paper questions as we always do. I hope that was useful for you as a recap, and we will do some questions in a moment. Okay, folks, so let's look at some of the past paper questions for this topic. Now, we'll have a look at this first one here. It says the diagram below shows an enzyme catalyzed reaction taking place in the presence of an inhibitor. Okay, so which line in the table identifies the molecules in the reaction? Right, let's have a look. So we've got P, we've got Q, we've got R, S, and T. If we have a look on the left-hand side here, we have R, which on the right-hand side seems to bind to the enzyme, but it binds away from the active site. So therefore, it's not going to be the substrate, it's not going to be the product, it must be the inhibitor. So R must be our inhibitor, okay? So it must be this one or this one here for the inhibitor. Now the substrate is what binds to the active site initially, and the product is what is made at the end of the reaction. And again, just from looking at the molecules here, it looks as though P is going to fit the active site on this side where we find the substrate, and that T and or S are going to be our products on the second side of the reaction, the product side there. So we're thinking that P will be our substrate and that T and S will be our products. So it's going to be C for the molecule. We can see the various shapes on them, which will be important in a second. Which row in the table shows the possible shapes of two molecules that could inhibit the enzyme above? Okay, so a competitive inhibitor binds to the active site. Now, by looking at the substrate and the shape of the substrate, we know this is the active site down at the bottom of the enzyme here. Okay, so is there any competitive inhibitor that could bind to the active site there? Well, that one probably could, that one probably could, that one couldn't, and that one there also couldn't. Similarly, because that's the active site, this must be the point where the inhibitor, the non-competitive inhibitor binds. So which of the non-competitive inhibitor shapes? So this one here would fit on the non-competitive site, and this one here as well. And that leaves us with an answer of B as the only one with both of them correct. Okay, the diagram shows a metabolic pathway that's controlled by end product inhibition. That's from an old paper. That's the same as our feedback inhibition that we just learned about. So for substance 4 to bring about end product inhibition, which of the following would it interact with? Well, we learned from our diagram that we looked at a moment ago that it would come back around here and it would inhibit enzyme 1. Okay, number 11 shows a metabolic pathway that's controlled again by that's the same question, isn't it? This substance here comes back and it inhibits enzyme 1. Okay, so that's going to be that one there. Metabolic pathways can be controlled by feedback inhibition, where an end product binds to an enzyme found at the start of the pathway. That sounds good. End product binds to an enzyme at the end of the pathway. Nope. Enzyme binds to a substrate found at the nope. And enzyme binds to nope. So it's going to be A. Okay. Number 7 here, as part of a metabolic pathway, substrate X is converted to product Y. Under what circumstances would this be reversed? Okay, so if there was an increase in both X and Y, if there was a decrease in the concentration of both X and Y, if there was an increase in the concentration of X and the removal of Y, and if there's an increase in the concentration of Y and the removal of X. So it's going to be that last one there, because if we have too much product, we would want to be reversing this reaction to get some more of the substrate. So it's going to be D for this one. Number 8, non-competitive inhibitors affect enzyme action by altering the shape of the active site. That sounds about right, but we'll hold off for now. Altering the shape of the substrate molecule? Nope. Competing for the active site? Nope. And competing for the substrate? Nope. So the answer is going to be A for that one. It's the only one that makes sense for non-competitive. Okay, so that's enough for the multi-choice just now. Let's have a look at a section 2 question. So trypsin is an enzyme which catalyzes the breakdown of proteins in the small intestine. The graphs below show how pH and temperature affect the activity of trypsin. So effect of pH and the effect of temperature. So before we get to the questions, I always have a quick look at the graphs and I think, okay, pH, so this is the activity. So that's the peak of the activity. So the optimum pH is 8 there. The optimum temperature is going to be around about 38 degrees, round about here. Okay, at this point here. And, you can see the usual as these go up, we can see an increase in enzyme activity, and then afterwards it goes back down as it begins to become denatured there. So anyway, just typical graphs as we'd expect for pH and temperature, but always good to have a look at them. So what's the optimum conditions for them? Well, we see that in the graph there. So we've got pH of 8 and we've got a temperature of, it's 38, I think. I mean, that graph is pretty difficult to read, but it looks like 38 to me. Okay, state the conditions over which trypsin shows at least 40% of its maximum activity. So the range of conditions. So 40% starts here and here on this graph, okay? That's 40% there. And over on this graph, it starts here and here, okay? So what we have to do is say that its pH is between, that's 6.6 and 8.8. So pH is 6.6 to 8.8. That's the range. The temperature, don't you guys write temp in the exam, but write the full word temperature. It goes from 20 degrees all the way up to 54 degrees. I think I'm reading that as 20 to 54 degrees. Okay. So that's the range of conditions there over which trypsin shows at least 40% of its maximum activity. Okay. Explain the role of the active site in enzyme catalyzed reactions. Okay, now this is a 3-marker. So the kind of things that it's looking for here. Let me get a text box so we can type it. Right. So the role of the active site. So we know that the active site is complementary to a specific substrate. We know that the active site will change shape to better fit the substrate. This is called induced fit. We know that this lowers activation energy, and we also know that the substrate has a high affinity for the active site, and the products have a low affinity for the active site. Okay, so something along these lines would get you the marks for that kind of question. It's basically just reciting what's in your notes in that induced fit section of the topic there, and that'll get you your 3 marks. Okay, don't worry, we're not going to do the big 10-mark question there for this topic, but hopefully we have gone over enough there that you're starting to understand this a little bit better. The other kind of questions you can be asked in this topic are things like compare competitive with non-competitive inhibition, or compare anabolic with catabolic reactions. So those might be 2 good areas to study up on and to have a go at doing a comparison question where you compare the 2 of them, because that might well come up in class tests or exams as well. Okay, folks, so I hope that was helpful again for this lesson. Let me know if you get any suggestions or if there's anything else you'd like me to go over. But for now, folks, class dismissed.